The topic discussed here is haploid plant production by means of androgenesis, anther and pollen culture. Haploids are individuals or tissues with somatic cells having gametic chromosome number. Doubling of these gametic chromosomes using certain mutagens like colchicine lead to production of diploid homozygous pure lines. In plant breeding, pure lines are of great importance in any improvement program. Haploids are formed in vivo spontaneously by distant hybridization techniques or by parthogenesis. These techniques are however rare and take generations. In vitro techniques were developed to shorten the time frame to achieve pure breeding lines. The two methods to produce haploid lines in vitro are androgenesis and gynogenesis. Androgenesis is a technique for production of haploid plants by means of pollen grains. The basic principle utilized in androgenesis is the haploid nature of pollens and the totipotency of microspores. Indian scientists Guha and Maheshwari are credited with the discovery of creating haploid lines by pollens of Datura. Presently, this technique has been utilized for over 200 species of plants, including economically important plants like rice, tomato, tea, barley. Androgenesis can be achieved by two ways based on the explant used for inoculation, anther or pollen. In anther culture, anthers at precise stage of development are excised and inoculated in nutrient media to give rise to haploid plants by organogenesis or embryogenesis. In case of pollen culture, uninucleate pollen grains are inoculated on nutrient media to develop into haploid plants by direct or indirect method by organogenesis or embryogenesis. In anther culture, intact anthers at precise developmental stage are inoculated into nutrient media. The pollens inside the anthers grow into haploid plants. Care should be taken as to not to damage the anther tissue because this would lead to formation of callus, which may in turn lead to indirect organogenesis to form a diploid plant from the anther tissue. The figure on the right illustrates the method of anther culture to get haploid plants by direct and indirect organogenesis, and also getting homozygous diploid plants by mutagen treatment of haploid cells. Steps for anther culture. Young flower buds are collected from healthy mother plants. The bud stage should be standardized based on size. The idea is to get microspores at precise stage of division. This can be estimated by staining techniques. Microspores at uninucleate stage are best preferred for induction of androgenic haploids. The buds are then surface sterilized and inoculated on nutrient media. Figure 1 shows the developmental stages of a microspore. The uninucleate stage, figure C is the best suited stage to induce androgenesis by giving external stimuli. The uninucleate cell undergoes asymmetric division to form a generative cell and a vegetative cell. The generative cell further undergoes mitotic division to form two male nuclei, while the vegetative cell accumulates nutrients like starch for pollen maturation. If the vegetative binucleate pollen starts storing starch, it stops androgenesis, or androgenesis cannot be triggered. Therefore, stress treatments are generally given at mid or late uninucleate stage, which triggers the microspore to enter into androgenesis phase. Stress can be given in the form of starvation or osmotic stress like incubating the anthers in mannitol solution. It could be heat shocks and cold shocks depending upon the plant used. The second figure shows the different stages of microspore embryogenesis. The figure here shows development of haploid plants by direct or indirect organogenesis. Haploid production by means of pollen culture. There are two ways of extracting or isolating pollens from anthers for haploid culture. First, squeezing method, and second, float culture method. In squeezing method, the young flower buds that are excised from the mother plant are surface sterilized and the anthers are placed into 20 ml of medium. These are squeezed gently with sterilized glass rod to separate the pollens from the anther walls. The prepared solution is then filtered through a suitable pore size mesh. The filtrate is centrifuged and the resulting pollen dense pellets are collected and washed. The final concentration of the pollens are adjusted to 100 to 104 pollens per ml. This is then inoculated into the nutrient media for generation of haploid plants. Float culture. In this technique, the anthers excised from the sterilized young buds are placed in a Petri plate containing the liquid medium and left for a few days. The anthers float in the liquid medium and release their pollen into the medium in few days. This is then centrifuged and inoculated into nutrient media. Certain other methods that are used to release or isolate pollens from anther or flower buds are magnetic bar stirring method, maceration, and blending. Magnetic bar stirring method is used in cases where the anthers cannot shed the microspores effectively in the liquid media. In such cases, a stirring force is applied using magnetic bar to release the microspores. Maceration. In this technique, the young flowers or the anthers are macerated or crushed using mortar and pestle. The resultant homogenate is then passed through filter paper to separate the microspores from the somatic tissue and then centrifuged using density gradient to separate the different stages of microspore. Blending technique. In this technique, anthers are suspended in mannitol solution and blended at high speed using microblenders. The suspension is then filtered through nylon papers or mesh of appropriate size to separate the microspores from the somatic tissue. This technique is efficient as it causes less damage and higher embryonic capacity and reproducibility is observed. However, the method needs to be standardized as to at what velocity and what time the blending to be done. The culture media plays an important role in induction of haploids from anthers and pollens. The nutritional requirement of pollens or microspores are different from anthers as certain factors required for induction of androgenesis are provided by the anther tissue. Therefore, it needs to be provided through the nutrient media. Formulated media like Morache and Skoog's media, Gamberg's media, niche media, etc. can be used for anther and pollen culture. In case of pollen culture, liquid media is preferred, while for anther culture, agar solidified media is used. Pre-treatment of anthers like starvation, osmotic stress, cold or heat treatments stimulate androgenesis. Nitrogen is found to be an important component for microspore culture. It is provided in the nutrient media in the form of amino acids. Besides these, certain important ingredients like glutamine, L-serine, and inositol, along with high concentrations of sucrose, are found to be favorable for haploid production. In certain cultures, it has been reported that a period of dark phase initiates the process of androgenesis. In certain cases, nurse culture techniques are utilized for anther and pollen culture. It was first developed in case of tomato. In this method, the anthers that are isolated from sterile young buds are placed horizontally on a semi-solid nutrient media. A filter paper is placed on top of it. Next, another set of pollen suspension obtained from a different anther is prepared in a liquid media and placed over the filter paper. This is incubated at 25 degrees centigrade in light conditions. Within a month, several haploid clones were observed in case of tomato. The figure on the right shows an illustration of nurse culture technique in tomato. Factors affecting androgenesis. The genotype of the mother plant plays an important role in inducting androgenesis. Certain genotypes have been reported to give large number of haploid plants. For this, the mother plant needs to be screened first and then selected. The physiological condition of the donor plant also matters. the selected mother plant should be healthy. Generally, young flowers at early season are used. Stages of microspore development, pre-treatment, culture media, temperature and light all affect androgenesis and has been discussed in previous slides. In certain cases, it has been found that charcoal induces androgenesis. It should also be seen while inoculating that the pollens or the anthers do not overcrowd the media. The figure here illustrates the generation of diploid homozygous lines by means of pollen and anther culture. Applications of pollen or anther culture. One of the major applications of pollen and anther culture is production of haploid culture lines in a very short period of time in large numbers. The conventional breeding methods used many generations of inbreeding and backcrossing to get haploid lines for improvement programs. Single pollens can be utilized for protoplast isolation. Haploid lines can also be used to obtain transgenic plants. It is used in many crop improvement programs. Pollen cultures avoid the risk of chimera and callus formation from anther walls. Anther and pollen cultures can be utilized in basic research for cytogenetic studies, to compare heterozygous diploid with haploid or homozygous diploid population. Recessive phenotypic characters can be identified very easily. It can be used to study genetic recombination in higher plants. It is used to study meiotic behavior of haploids on duplication within a species and understand the phylogenetic relationship between species. Haploids are used in the production of monosomic, nullisomic, and other aneuploids. Genetic analysis could be performed on haploid population to establish inheritance patterns. The disadvantages of pollen or anther culture are it requires skill to remove anthers without causing damage. The stage of microspore development too needs to be standardized. Not much success has been obtained in case of cereal crops. In case of anther culture, risk of chimera and callus formation is present. Advantages of pollen culture over anther culture. First, there is overcrowding of pollen grains in anthers. In pollen culture, elimination and isolated pollen grains are equally exposed to nutrient media. Second, unwanted growth of diploid cells from anther wall is eliminated. Stages of androgenesis can be manipulated and observed. Pollens are directly transformed to embryos, hence suitable for understanding biochemistry and physiology of androgenesis. The pollens are ideal for transformation and mutagenic studies. The pollens can uniformly be exposed to chemical and physical mutagen when compared to anther cultures. The production of homozygous diploid plant from pollen culture or anther culture derived haploids can be done by colchicine treatment, endomitosis, or by fusion of pollen nuclei. Thank you.